Ever wondered why some cancer treatments just stop working, even when everything seems to be going right? The answer might be hiding deep inside the DNA of those tumors. So deep, in fact, that standard tests completely miss it. Let's talk about a breakthrough computational tool called BACDAC, developed by researchers at the Mayo Clinic. This tool is uncovering hidden DNA changes that could be the secret culprits behind aggressive cancer growth and treatment resistance. So what makes these DNA changes so hard to catch? It comes down to structural alterations, big, sweeping changes in a tumor's chromosomes that regular tests can't always pick up, especially if the tissue sample is tiny or a bit degraded. Think of it like trying to find a typo in a book but only being allowed to see a few words at a time. Not so easy, right? Here's where BACDAC comes in. This tool reads the entire genome, even if the sample isn't perfect. It's designed to spot signs of genomic instability, which is basically when the DNA inside cancer cells is a chaotic mess. And that chaos can be a warning sign. It means the tumor might be more aggressive, or more likely to resist treatment. At the center of BACDAC's approach is something called ploidy. In simple terms, ploidy is the number of complete sets of chromosomes in a cell. Normal human cells have 2 sets, but cancer cells can have more, or sometimes less. If a tumor has doubled its entire genome, that's called whole genome doubling. And guess what? That's usually a bad sign. It's often linked to faster growing, harder to treat cancers. In a recent study, BACDAC was used to analyze over 650 tumors across 12 different types of cancer. The tool caught tons of these whole genome doubling events, genetic shakeups that would have been invisible otherwise. But BACDAC doesn't just crunch numbers. It actually creates a visual summary, a constellation plot, that maps out whether a tumor's chromosomes are stable or totally disrupted. It's like getting a weather map for cancer's DNA, making it much easier for researchers and doctors to interpret the results. Dr. George Vesmatsis, one of the lead researchers, summed it up best. This tool lets us see a layer of the genome that's been invisible until now. After decades of research into how DNA instability drives cancer, BACDAC is finally giving us a way to spot these changes across thousands of tumors and possibly in real clinical settings soon. The next step, the Mayo Clinic team is working to make BACDAC a tool that pathologists can actually use to guide treatment choices. If we can see how a tumor's DNA is changing, we might just stay one step ahead of cancer's tricks. So, the next time you think about how technology is changing medicine, remember, sometimes the biggest breakthroughs are hiding in the smallest details.